I'll just give it a few seconds to let people filter in. All right. Hello, everyone. Good morning, good afternoon, and good evening, wherever you are. Welcome again to the Long-Term Animal Research Seminar Series. We continue this semester set of seminars with talks from Dr. Lauren Petrullo and Dr. Michelle Brown. And before I introduce our speakers, I'll make a couple of announcements. First of all, we are evaluating the effectiveness of this series, so we need your help. So please take a few minutes to fill out the survey that's posted at the top of the Twitter page, and which I'll add to the chat in a few seconds. And so this evaluation is really a critical component of our broader impacts, and the results from this survey will help us chart our path forward. So we really appreciate your time in filling out how you feel about the LTAR series. Secondly, if you're participating via Zoom, you should see a Q&A tab at the top or bottom of your screen. If you open that tab, you'll be able to type any questions that you have, as well as see and upvote other people's questions. At the end of the talk, we'll go through those questions, starting with the ones with the most votes. So please use the Q&A tab. Lastly, recordings of the talks will be available on YouTube shortly after they conclude. So if you need to leave early or know others who are unable to attend live, this talk will be available for viewing and reviewing after it's complete. Now that business is taken care of, I'm pleased to introduce our first speaker, Lauren Petrullo. Lauren received her PhD from Stony Brook University before starting her current position as an NSF postdoctoral research fellow in the Department of Psychology at the University of Michigan. Lauren's work focuses on the mediating effects of hormones and microbes on the relationship between environments and phenotypes. She has worked on maternal transfer of gut microbiomes in vervet monkeys and the effects of early adversity on phenotypes. Her current work uses a combination of long-term observational data, experiments, and physiological measures to test hypotheses about how offspring are primed to respond to changes in the environment in squirrels. So I'm really excited to introduce Lauren and hear more about her work today.
Okay, hopefully this is good. Thank you, Liz and Matthew, for that really nice introduction, and good morning, everybody. My name is Lauren. I'm currently a postdoc at the University of Michigan, where I'm working with Ben Donser as part of the kluani red squirrel project. I just recently completed my PhD this past August, as Liz said, so I began this postdoc just a couple months ago, so please do bear with me as I take you through some of this preliminary data. So at present, there is a pretty comprehensive body of literature that demonstrates that changes to an animal's environment can result in changes to that animal's gut microbiome, which is the collection of bacteria that live inside of the gastrointestinal tract of a host. And we see these effects occur in all corners of the animal kingdom, from fish to birds to reptiles to mammals, including humans. And as biologists, we care about this relationship because the gut microbiome can itself exert influence over many different aspects of host physiology, including things like immune function, brain development, metabolism, and behavior. And these all have implications for host life history and fitness. But as we uncover more and more about environmental effects on the microbiome, two big questions remain. First, how do these changes occur, and what are the mechanisms that link the environment to the gut microbiome? And second, why do these changes occur, and can they be adaptive in the short or long term? So today I'm going to focus on only one of these questions, the how. One potential way that the environment can result in changes to the gut microbiome is through host production of glucocorticoids, which are metabolic hormones that are involved in energy regulation and the host stress response. So data from experimental rodent models has shown that chronic and acute laboratory stressors, so things like isolation, light, and noise stress, can result in changes to the composition of the gut microbiome. And these changes are assumed to be maladaptive for hosts in that they destabilize the community in such a way that can allow for increased susceptibility to infection and to pathogens. But in terms of wild animals, I know I don't have to sell you all on the utility of glucocorticoids, but GCs are a valuable integrative measure of an animal's ecology because they vary in response to changes in things like the social environment and food availability. And although we know that GCs are linked to the microbiome in the lab, very little is known about the relationship between GCs and the microbiome in wild animals that are exposed to natural challenges. But there is a little bit that we do know, and it comes from just a handful of studies in birds, fish, and squirrels that have demonstrated a pretty consistent negative relationship between glucocorticoids and microbial diversity, such that high levels of glucocorticoids are associated with low community diversity. And diversity is an important trait of a microbiome because it contributes to microbial stability and the resilience against things like pathogens, because more niche space is occupied by a more varied assortment of taxa. So if we look here at these 3 mock microbial communities, each shape and color is a different species. And we can see in the first community that there are only circles and triangles, but there's many different colors or species of each. So this community would be considered taxonomically rich. The second community is both rich and even because the spread of these species is consistent and no one species appears to be dominating the entire community. And finally, this last community appears phylogenetically diverse because it has some taxa, like the squares and stars and these arrows, that look very distantly related to the triangles and circles. And it's generally considered optimal to have greater microbial diversity in all 3 measures because you can imagine the empty space in a non-diverse community might lead to susceptibility to infection by pathogens. But overall, we really need more data from wild animal populations to figure out whether GCs and the microbiome have a relationship that's actually conserved across species. What different factors might modify this relationship and what effects a reduction in microbial diversity might actually have on animals. So I'm working now in collaboration with the Kluani Red Squirrel Project, which is a long-term interdisciplinary field project that has been going on since 1987 and is focused on studying the ecology and evolution of red squirrels living in Kluani National Park in Yukon, Canada. And this population of red squirrels is an excellent system for trying to disentangle the relationship between glucocorticoids and the microbiome for a number of different reasons. So from a practical perspective, all squirrels are marked shortly after birth with metal ear tags and combos of colored wires for easy identification during observation and during trapping. The squirrels also don't disperse very far, which allows for individual data collection from birth through death, including things like recruitment into the breeding population, reproductive effort, and lifespan. From an ecological perspective, these squirrels deal with enormous environmental changes. So first, the availability of their primary food source, white spruce cones, exhibits profound fluctuations such that mast years, whereby an abundance of cones are produced and indicated here on this plot by the arrows, are followed by multiple years of little to no cone production. And on top of this, there's no new food to eat at all from September 1st to June 1st. And so squirrels have to rely on the food that they've hoarded in their territories previously to hold them over, and therefore are understandably quite territorial. In addition to fluctuations in food availability, red squirrels also deal with profound fluctuations in both conspecific density as well as predator abundance. And so constructing a framework for understanding how and why glucocorticoids may impact the gut microbiome in a population like this looks something like this. So in this population, fecal glucocorticoid metabolites are known to integrate 3 major ecological challenges, and in turn might alter the gut microbiome composition in such a way that it impacts host life history and trade-offs between these life history traits. So for example, a decrease in microbial diversity as a result of high glucocorticoid concentrations might reduce survival by increasing host susceptibility to pathogens. So for the sake of this talk, I'm just going to focus on addressing one research question in particular. Do glucocorticoids predict gut microbiome composition in wild red squirrels? So in line with prior studies, we predicted to find that microbial diversity would be negatively associated with GCs, such that higher GCs would predict lower levels of gut microbiome diversity. And we also predicted that changes in GCs would be associated with changes in the relative abundance of some important genera that are present in the red squirrel gut microbiome. So if GC-induced shifts in the gut microbiome community are expected to alter host physiology in some way, we might expect that changes would happen to these major players in the gut community. So to address these questions, we used 227 fecal samples collected from 88 individuals that were collected as part of a previous published microbiome project by Wren et al. From 2017. These same samples were also assayed for fecal glucocorticoid metabolite concentrations. And the samples were collected during 2008, 2009, and 2010, with 2010 being a masked year. So for the microbiome people out there, we sequenced the V1, V3 region, and sequences were resolved to OTUs at about the genus level using a 97% similarity threshold. From a statistical perspective, the linear mixed models we constructed were used to test whether fecal glucocorticoid metabolite concentrations predicted microbial diversity, and negative binomial mixed models were used to test how FGM predicted the relative abundance of specific bacterial taxa. All of the models controlled for age, season, sex, whether the individual was part of any additional experiments, and included individual ID as a random effect. So in line with our first prediction, as well as with work done by prior researchers, we found that a significant association between fecal glucocorticoid metabolites and community diversity was actually negative, such that higher levels of FGM were associated with lower community diversity across all three measures of diversity. And if we look at the trajectory of microbial diversity across the sampling months throughout these 3 years, we can see that diversity takes a trajectory characterized by recurrent dips and peaks. And the dips in diversity that occur from early spring to about June are consistent with the consumption of primarily hoarded cones from earlier years. Once June rolls around, most green foods become available and the squirrel's diet becomes much more heterogeneous, with the incorporation of things like spruce buds, fresh cones, and fresh mushrooms. So they have an overall very mixed diet of fresh foods that peaks in July. Now, if we map changes in fecal glucocorticoid metabolites over time onto this plot as well, we can see that microbial diversity and glucocorticoids pretty strongly oppose each other across 2008 and 2009. However, in 2010, the relationship appears somewhat different, with microbial diversity beginning to track changes in fecal glucocorticoid metabolites starting about in April. And it appears that the relationship between FGM and the microbiome that we see in the previous years of 2008 and 2009 is decoupled in the masked year of 2010, such that FGM did not significantly predict any measure of microbial diversity during this year, suggesting that something funny might be going on with this relationship during masked years. Second, we predicted that FGM would be associated with changes in the relative abundance of some key resident microbiota. So listed here inside of this yellow oval are 10 core genera, which means that they're present in every sample across individuals, time, and space within this data set. However, what we found was that changes in FGM were mostly associated with changes in the relative abundance of non-core taxa, with the exception of increases in Clostridium and Oscillospira, indicated here. So this figure is showing bacterial genera that change significantly with FGM concentrations. On the y-axis here are the genera, and on the x-axis are the model estimates, or essentially effect sizes. So genera that have negative model estimates exhibited a decrease with increased FGM, while genera with positive estimates increased alongside FGM concentrations. We also found that higher FGM concentrations were associated with a reduction in some pathogenic taxa, such as Yersinia. And very broadly, we find that an increase in FGM was associated with a decrease in environmental bacteria. So for example, bacteria that are largely found in freshwaters and soils, and an increase in bacteria involved in host metabolic function. But I want to point out that many of these taxa, especially the ones located at the very top and the very bottom, are really unknown in terms of their function and their roles as members of the gut microbiome. So to summarize, overall, we found a negative relationship between FGM and the gut microbiome. This is in line with previous studies on wild animals. We also found that the relationship between FGM and the gut microbiome was decoupled in 2010, in that year where the food resources were super abundant. We also found a decrease in pathogens and environmental bacteria, as well as an increase in metabolic bacteria very broadly, with an increase in fecal glucocorticoid metabolites. And so some of the main take-home points here are that ecology really matters when we consider how the microbiome and host physiology interact. So we know that some weird stuff is going on during these mast years in this population. So for example, there are shifts in reproductive behavior, and these types of things could be contributing to the decoupling of this relationship that we see. And these data also suggest, as does a previous study on birds, that there may actually be a protective effect of what we call stress on pathogenic infection within the gut microbiome, but we would really need finer resolution at the level of a bacterial strain to know for certain. But overall, given these findings that are very preliminary, we need to incorporate some more detailed ecological data, some experimental data holding some of the drivers of variation in FGM constant, and some functional microbiome data to get a better idea of what's going on here. Because the conceptual model that I showed you at the beginning of this talk probably actually looks something more like this, much messier, with multiple potential connections between these various components, including a potential bidirectional relationship between GCs and the gut microbiome. So again, these data are preliminary, and they're just the first stepping stones towards some bigger questions I hope to address in the coming years as part of this project. But thank you again to the seminar organizers for inviting me, to my lab and collaborators, the KRSP, and everyone for joining in.
Great, thanks, Lauren. That was really awesome and interesting. All right, so we have a question in the chat from Mark Hauber, who asks if there's a sex difference in FGM and GMB during the mating season or anytime.
Yeah, so there is no sex difference in microbiome composition that we detected, and that's pretty consistent with what we see in other animals. We don't see very many sex differences in microbiome composition, but there are differences in glucocorticoid production between the sexes during the mating season. So unfortunately, the sampling time for these 3 years just kind of ends a bit too early, especially in that masting year. We only get to about June or July. So what we're planning on doing is looking at the glucocorticoid data beyond those months in 2010 to get an idea of what's going on.
And do you have repeated measures on individuals, is that right?
Yep. Yeah. So it's between 2 and 3 samples per individual.
Okay.
Yeah.
Maybe not enough, but I was curious if you see any individual differences in the plasticity of the relationship between glucocorticoids and gut microbiome.
Yeah, so parsing apart kind of the among individual versus within individual variation in this relationship is kind of my next step. So I'll keep you posted.
Cool. Looking forward to it. All right, and I think Matthew has a question too.
Yeah.
Thanks. So the difference in the relationship in 2010, I think, is really interesting. Does that suggest to you that when GC levels are not perhaps the result of food-related stress, but some other kind of stress, that those things are affecting the microbiome differently? And is there support for that idea in the captive literature?
So I've been thinking this too. There's unfortunately no support for that in the captive work, just because most of the captive work is purely experimental rodent models, usually germ-free models. But yeah, it definitely suggests that potentially there's some ecological factor decoupling this relationship, or that the drivers of variation in the gut microbiome are different than the drivers in variation in fecal glucocorticoid metabolites.
Thanks.
Yeah.
All right, so I think we're going to move on. Thank you, Lauren, so much for that interesting talk. And so now I'm really pleased to introduce our second speaker, Dr. Michelle Brown. Michelle is an assistant professor in the Department of Anthropology at UC Santa Barbara. And she's also the co-director of the Biobehavioral Lab at UCSB, a research affiliate at the Max Planck Institute of Animal Behavior, and the founder and director of the Mgogo Monkey Project. Her group uses observational data on movement, foraging, communication, and agonism of 5 different focal primate species to understand the causes and consequences of competition. Currently, her work is focused on how strategies used in feeding competition drive social variation. And the comparative nature of Michelle's work gives really important insights into how different species respond to similar environmental pressures. So I'm thrilled to welcome Michelle today to hear about the costs and benefits of intergroup conflict.
Thank you, Liz. Is everything good? Can you see my screen?
Looks great.
Okay, super. So as Liz mentioned, I'll be talking about intergroup conflict today. And my focus on this topic stems from the body of theory that we have that indicates that these are fairly important conflicts within social animal lives. So these conflicts can determine an individual's access to resources, both in the short term and the long term. And then ultimately, there's a sort of feedback loop where a strong degree of between group contest competition can modulate within group social relationships such that individuals need to cooperate more explicitly with each other in order to effectively defend group resources. Now, in terms of understanding the dynamics of these conflicts, there's a huge diversity of patterning that we see across species and even within species over time and over populations in terms of the context in which these intergroup encounters are happening, the resources that are being defended, and the dynamics of these particular conflicts as they escalate or in some cases fail to escalate. So we use evolutionary game theory of animal contests to understand the patterning of these conflicts. And this body of theory is relevant not just to contests between individual animals, but also between social groups. And the idea here is that in order to explain who initiates a contest, who then escalates it to higher and higher degrees or intensities of aggression, and ultimately who wins, we have to look at asymmetries in two particular factors to understand or to answer these questions. So these asymmetries could be in terms of motivation of the contestants, and that motivation stems from the payoffs that are the resources that they are defending or seeking access to. And also asymmetries in terms of the power of the contestants or their resource holding potential, RHP. So this might be their ability to monopolize those resources. These asymmetries are determined by, in some cases, conventions. One such convention is ownership on occasion. We see this in some species where the owner of a resource or the owner of a territory might be more likely to win versus a newcomer. And other times what we see is a pattern of assessment. So as the interaction progresses, the two parties are assessing each other's motivation and assessing each other's resource holding potential. Although there's quite a bit of debate about the nature of assessment and who's assessing what. And so we'll get to that a bit later. Now, in going into the primate literature on intergroup conflicts, there was a lot of conflicting approaches that I saw. And so my first step was to try to create a framework to organize my studies on this topic. And so first we start with the predictors of when individuals are going to be initiating, escalating, and winning these encounters. And I say individuals, but I also mean groups here. And so there's three general categories of predictors. There's again, the resource holding potential and the payoffs that I mentioned on the previous slide. And for groups, the resource holding potential could be numeric odds. Is this group larger than the other group? Or in terms of the number of actual participants, or sometimes even the cumulative strength of those participants. In terms of the payoffs in primate societies, typically the resources that are being defended are food and or mates. On occasion, there's other types of resources, but food and mates are really the most common resources in dispute. In terms of individual behavior and whether those individuals participate or defect from a particular encounter, we can look at disposition-related predictors, so the rank of the animal, their degree of relatedness to group mates or even to members of the opposing group, their age or tenure in the group, for females in particular, their reproductive phase, or the personality of some individuals and their reactivity to social conflict. The scope of responses that we examine could be in terms of individual animals. It could be females versus males or the group as a whole. And then ultimately, there's the phases of the contest. So for those species that communicate between groups using long distance vocalizations or even scent marks, we can look at the group's movements in response to those spatial or temporal cues. And then there's another phase oftentimes that we see in terms of initiation. This is a brief window of time where the groups are not yet able to see each other necessarily and are certainly not close enough to have physical contact. But they can hear each other's short-range calls, and this window of time gives them an opportunity to decide, are they going to approach to truly initiate that encounter, or are they going to retreat? Once they are within visual and sometimes even physical contact, what are the patterns of escalation? So agonism or affiliation. Now in the species that I study, there's no affiliation, so I'm not going to talk about that anymore. And ultimately, what is the outcome of those interactions? So this framework guides my approach to understanding intergroup conflicts, especially in primates. I'm very interested in measuring food. This, again, is one of the main types of resources that are contested among primate groups. And on the surface of things, it should be relatively straightforward. So food should be defended when it is economically feasible to do so. So when access to food resources limits reproductive success and those food resources are monopolizable, so a group can effectively exclude another neighboring group from that resource. In practice, it's a lot more challenging to measure food availability than this sort of simple setup here. And in primates, this is exacerbated by the fact that they have diverse and flexible diets. So primates tend to be fairly omnivorous. So even though they might have a preference for fruit or a preference for leaves, there's quite a number of other items that can be mixed in. So for the species that I study, they do prefer to eat fruit, but they'll mix in some young leaves, some flowers, and certainly quite a lot of insects. It can be very challenging to measure some of these food items, especially the insects, on the spatial scale of a home range. And frankly, we haven't quite gotten a good grip on how to do that just yet. But in terms of plant-based resources, there's a lot of variation in terms of the types of characteristics that different researchers measure, especially in the context of food competition. And I've divided these into two general categories of regional characteristics, meaning across the scale of the home range, versus local characteristics, which are specific to the area where the two groups have encountered each other, and I call that the contest site. So the regional characteristics can include the density or the abundance of those food resources, the distribution of feeding patches, the quality of those patches, whereas local characteristics tend to be about the intensity of use of that particular site, that location, is it sort of closer to the center of the home range or towards the periphery, the size of the particular food resource that might be consumed at that time, and the degree of depletion of that resource or the satiation of the group. And unfortunately, most research on between group conflicts and testing of food defense tends to look at only one or two of these predictors rather than the entire suite of possible predictors. So moving forward, I really wanted to take a comprehensive approach to understanding food defense in my study system. So I chose to look at two particular species. These are gray-cheeked mangabeys and red-tailed monkeys, and I work at the Ngogo site in Kibale National Park in Western Uganda. I chose to work there because it has fairly high densities of both of these species, which allows me to have a decent sample size of observations of intergroup conflicts for them. And I chose these two species because they have such similar diets. And the idea based on primate socioecological theory is that if they're consuming similar diets, then they ought to be defending those food resources or their access to those resources in similar ways. And that is not at all what we see. Mangabeys rarely have intergroup encounters, or at least the frequency of those encounters is much, much lower than what we see in red-tailed monkeys. And I'll talk a little bit more about some of those differences in the next few slides. Now, there's a lot of things that these two species share in addition to the diet. So they have overlapping home ranges, they're often traveling together to the same food resources, and they have similar group sizes and similar travel distances per day. But there are a few important differences between them as well. So mangabey groups tend to have multiple males, whereas red-tailed groups have only one male. Mangabey home ranges are about 5 times larger than the size of a red-tail home range, and mangabeys are also larger bodied. But aside from that, again, there's many important similarities, including the fact that females in both species are philopatric, so they stay in the groups where they are born. So looking at these different contest phases across the two species, I start with group movements in response to long distance vocalizations. And this analysis is restricted to gray-cheeked mangabes because their long distance vocalization can be heard from the center of one home range to the center of the next home range. And that is not true for red-tailed monkeys. The loudest vocalization that the red-tails produce can really only be heard maybe 200 meters away, plus or minus some. So for this analysis, I combined my observations from Ngogo, which is a high density population of mangabes, with the data from Peter Wazer, who had studied the same species in the same national park, but at a site that's just about 12, 15 kilometers away. And he conducted his analyses in the 1970s. So it's quite a time gap between our two studies, and there were some differences in terms of our methods, but we were both observing groups and how they respond to neighbors. We examined group travel responses in the 30 minutes after hearing a long distance vocalization, either from a real neighbor or in response to a simulated neighbor during a playback trial. And what we found is that, first and foremost, when groups hear a neighbor's call, for the most part, they tend to ignore it. Whatever they were doing before, whatever direction they were traveling beforehand, they continued in that direction. That actually makes sense because these groups are hearing the calls of their neighbors all day. And if they change direction, if they rush toward a neighbor every single time they heard that long distance vocalization, they wouldn't get a whole lot done. All their feeding bouts would be interrupted. It's a very inefficient strategy. So first and foremost, they basically keep calm and carry on. Now, there are occasions under which groups actually retreat from each other, and this matches up quite nicely with Wazer's original findings from the 70s, where groups actually seem to be avoiding each other most of the time. And so basically, if the groups were close to each other, the listening group retreated, and it had the net result of creating a spatial buffer between the groups. Now, again, I was studying a very high density population, and I saw quite a bit more intergroup conflicts than Wazer had observed. So I thought there must be some context under which groups are choosing to approach each other and to engage in these conflicts. And sure enough, if the calling group had entered the overlap zone, so the shared area of the two groups' home ranges, the listening group would approach. Now, that's not to say that they were starting an intergroup conflict, but they were approaching a little bit, and that gave them the ability to sort of monitor what was happening with that neighboring group in the overlap zone. Moreover, there's a second context under which groups would approach each other. And that's when, in short, males were distracted by the presence of fertile females. Now, this is actually a much more specific context where, given that there's multiple males in each mangabe group, there's a dominance hierarchy where there's one male who's high ranking, and then there's basically everybody else who's lower ranking, and it's not a clear linear dominance hierarchy. But typically, there's only one fertile female in the group at a time because these are non-seasonal breeders. And so that one fertile female is mate guarded by the alpha male. So when there's just one female, no problem. The group sort of continues in its normal travel path. But when there's two fertile females, meaning that they have sexual swellings that are maximally inflated, everything breaks down. That's because the lower ranking males suddenly have an opportunity to mate with a female who is not being guarded by the alpha male. And at that time, their attention seems to be concentrated on that second fertile female and not on the other members of the group. And what we see is that the group actually approaches the neighbors. So what I'm going to demonstrate to you in the next couple of slides is that there seem to be several instances in which females' interest appears to be in approaching neighbors, possibly to defend food resources, but very often their movements are constrained by male interests. So males are trying to keep them away from the neighboring group. Now in this analysis, there was no effect of group size or of food abundance, but I'll follow up on that shortly. Next, I looked at the pattern of initiation. So again, groups are fairly close to each other. They can hear short range calls. They can't yet see each other, and they have a decision to make. Are we going to approach or are we going to retreat? Now, I examined binary responses by females versus males. So is there any female who makes a strong approach to the caller or any male who makes a strong approach? And interestingly, the only food-related variable, the most important food-related variable that popped out in this analysis was the amount of time that the two groups spent in that area. So if the listening group had just arrived, they were likely to approach. And they were even more likely to approach if the calling group had been in the area for quite some time and had had a chance to satiate themselves on the resource in that area. So this really indicates to us that the Mingabe groups are not only paying attention to the neighbors, monitoring when they go into that overlap zone between the home ranges, but are also tracking how long those groups have spent in that area consuming particular resources, and that determines when they're willing to push out those neighbors. So this is a fairly unusual pattern that we see where there's assessment of resource value, both for the focal group and for the opposing group. Again, however, there was no effective group size, and that was a little surprising to me because we do expect to see asymmetries in resource holding potential, typically in terms of group size when we're talking about group level conflicts, playing a role here. And a paper had been published a few years ago by Sarah Van Bell and Clara Scarry, indicating that across many different species, not just primates, that there's several instances highlighted in yellow where we don't see a group size effect, when of course we would expect one to occur, but we don't see it. And I figure that there's probably two possible explanations here. One is that maybe the value of the resources to be defended, the payoff asymmetries, is so strong that it overwhelms the asymmetry and resource holding potential. And that's possible. But there's also a second explanation, sort of a more mundane explanation, which is that perhaps there's just simply different methods that are being used across studies that might explain this. So going back to my data on initiation of these conflicts in mangabes, instead of just looking at binary strong approaches or the absence of strong approaches, now I looked at focal follow data from individual animals and looked at all of their movement responses, whether it was 5 meters or 50 or 100 meters, and whether it was toward the caller or even away from the caller. So a much more fine-scaled analysis. And sure enough, there's an effective group size. So circled in blue are the smaller groups who tend to retreat from the caller, and circled in green are the larger groups that tend to approach the caller. Great, there's an effective group size, just as we would expect. However, in comparing those two levels of analysis, so the binary strong responses versus the continuous fine-scaled responses, I thought, well, what happens to a focal animal's response if they see that another individual in their group, especially another individual of the same sex, was making a strong approach? Other studies, for instance, on white-faced capuchins on Barro Colorado Island in Panama, indicates that we see some degree of defection. So groups or individuals are actually less likely to approach if someone else in the group has taken it upon themselves to deal with the opposing group. So do we see the same pattern of defection in mangabes? So looking at this question, first, the context where no individual of the same sex made a strong approach. And what we see is that the focal animals are tending to retreat from the caller. But if we see those contexts where a same-sex individual is making a strong approach, all of a sudden those focal animals are moving toward the calling group. So there's actually a cooperative tendency in gray-cheeked mangabes, which conflicts with what we see with the capuchins. And in fact, this effect is strong enough that it swamps out the group size effect. So this idea that resource holding potential should shape group responses to opponents is actually overwhelmed by cooperation in mangabes. Now you might say, okay, what about those red-tailed monkeys you mentioned? And unfortunately, it's not quite this as easy to do this analysis of initiation patterns here. And that's because most of the time red-tailed groups actually just seem to bump into each other as they're moving around in the habitat. And this is a process that is initiated by mangabes. So red-tailed monkeys love to travel with mangabes, so much so that you see the red-tail home range here in red, the polygon, and it's overlaid by the much larger yellow mangabe home range. So as the mangabes are traveling through their range, as they're approaching a red-tail group, the red-tails sit at the edge of their home range and wait for the mangabes. But the mangabes are traveling from red-tail range to red-tail range to red-tail range. And as they pass from one range to the next, those red-tail groups meet and they have an encounter. It's not necessarily an aggressive encounter, but they're having an encounter. So if we look at those periods per red-tail group and see whether or not they're traveling with mangabes, if they are with mangabes, 100% of the time, they're going to have an intergroup encounter. If they're traveling alone, 2/3 of the time, there's no encounters with other red-tailed groups, and 1/3 of the time, they will have an encounter. But again, there's no patterning in terms of whether they're aggressive in the presence or absence of manga bees. So it doesn't seem like they're actually fighting for access to the manga bees. So next, looking at patterns of aggression and analyzing explicitly whether there's evidence of food defense by males and females. In short, what I find from manga bees is that the male behavior, so male aggressive participation in these encounters, corresponds very closely with some of the predicted context under which food is thought to be limiting and monopolizable. And that is when overall abundance of food in the home range is low and the feeding patches are widely dispersed in the environment. So these are regional characteristics. And from red tails, both males and females, what we see is a correlation between aggressive participation and local characteristics. So yes, there is evidence of food defense, but why does each species respond to different characteristics? Now, again, across the primate literature, this is not unusual. One species will respond to one set of characteristics and another species will respond to another. So what drives that patterning? Now, again, thinking about these two particular species, manga bee groups don't encounter each other very often. It's maybe once a week, whereas red tails groups often encounter each other every day. So there seems to be a difference in terms of the frequency that they're encountering neighboring groups and the pressure that they experience from those neighbors. So I created an index of intruder pressure, which is the ratio of group density to home range size. So you can imagine that in both of these scenarios of low and high intruder pressure, there's the same number of groups in the area, but the home range sizes are different. And so when the home ranges are smaller, you might have less overlap and thus less meeting up of those groups, in contrast with larger home ranges where there's more overlap and more likelihood of just randomly encountering the neighbors. Sure enough, when we look across the primate literature, those species that have a high intruder pressure, a value of 10 or more, aggressive participation and food defense corresponds with local characteristics. Those that have a low intruder pressure correspond with regional characteristics of food availability. Moreover, when we consider that some species exhibit core area defense, where food resources seem to be concentrated in the core area, and groups are more likely to initiate and win conflicts in the core of their home range. Uh-oh, things froze up. Can you guys see me?
Yeah, we can see you.
Okay, something just happened on my end. I'm not quite sure. Okay. So core area defense.
We can't see your PowerPoint now, though.
Oh, okay. Let me fix this. Okay. How about now?
Looks
Nope.
great.
Oh, yeah, you can?
Well, we can see it. Yeah.
Okay. Everything has sort of swept around on multiple screens. Okay. So some species exhibit core area defense, others exhibit boundary defense where resources are actually more concentrated apparently on the periphery of the home range, and this is where they have encounters. And then in the middle, there's sort of this intermediate stage or intermediate pattern where we see no spatial bias in terms of core versus periphery, but just wherever resources are distributed in the home range as patch defense. Here we go. Uh-oh, come on. All right, I might have to reset this momentarily. Oh, wait a minute, sorry. Okay.
No problem.
Okay, can you see the screen again?
Yes, we can.
Thank you. Okay. Now, so there is evidence of food defense by both great-cheeked mangabees and red-tailed monkeys, but there's this sort of odd pattern where we don't see female mangabees participating in between group contests. And by all logic, we should be seeing them participating. Like red tails, females are philopatric, so they have the opportunity to cooperate with kin in the defense of shared food resources. And their food is clearly limited and monopolizable because we see males defending these resources. So I thought perhaps there's some aspect of male behavior that might be constraining females. And we do see that males sometimes herd females, keep them away from the neighboring group, but herding wasn't targeting fertile females. It was actually targeting every other group member who wasn't an adult male. And in fact, because there's these multiple males in the group, there actually seemed to be certain roles that males were playing, and their participation in a certain role varied from one encounter to the next. So these are not stable roles. So one male would be on offense, and he would run off to the neighboring group, chase individuals, and then come back to his group, catch his breath, rinse and repeat. Then there'd be the herder who would sit at the edge of his group, and sometimes he was herding, but other times he was merely kind of sitting there and glaring. So just going by herding behavior alone wasn't enough. It was more the presence of the herder and where his attention was directed that seemed to be relevant. And then there would be a third male sort of at the back of the group with any female who had unweaned infants. And I assume that he was probably the sire of these infants, but we don't yet have genetic data to test that. And females participated in about 20% of encounters, and it just so happens that that 20% occurs to those times when there's multiple fertile females in the group with maximally inflated sexual swellings. And again, this is a time when male attention is directed specifically at that second fertile females in terms of the lower ranking males, which frees up all the other group members to do what they would like to do, which apparently is to go and fight with the neighboring group. So we're seeing this pattern where males are constraining female behavior. And more generally, this is a pattern that occurs across the primate order. In a meta-analysis with a colleague, Chris Schmidt at Boston University, we're examining this pattern across different primate taxa, and this is holding up in a fairly robust pattern. Now, in terms of male behavior and defense of female mates, we do see that this shapes male responses, but in very different ways because of the difference in terms of within group mating competition, which is absent for red tails and present for mangabes. So for red tails, controlling for the value of the feeding site, whether it's a low value site or high value site, male participation actually increases in the presence of estrus females. In contrast, mangabe male participation decreases in the presence of estrus females, again, because of this need to make guard against other males within the same social group. And so thinking about the broader primate literature again, what we might expect to see is that it should be important in terms of the timing of these two types of resources, food and mates. So when only mates are defended, say perhaps in mountain gorillas, that might be limited to a certain time of year or several small periods. Then there might be species where there's separate periods of food being both limiting and defensible versus mates being limiting and defensible. And then here in the final situation, these are simultaneous periods of food and mate defense. So if we think about male participation, what you should see is that in this pattern here where there's low skew in male access to fertile females within the group, male participation should increase during these periods, regardless of whether they're defending food and mates and doing so at the same time. But if there's a pattern of high reproductive skew, as we see in mangabes, instead there should be a decrease in participation because of the need to compete against other males within the same social group for access to mates. Now here, if there's a separate period of food and mate defense, males might actually increase their participation in order to demonstrate their viability in this sense, because there's some idea that perhaps females could be choosing among males based on their ability to defend resources. And this is a pattern that across, say, about 12 different primate species holds up thus far. Unfortunately, there's not a whole lot of literature out there that looks specifically at the timing of food and mate defense across primate species. So, moving on to patterns of individual participation during these conflicts, what we see is And this is an analysis that is focused on red-tailed monkeys. This is something that I did a few years ago for my postdoc, looking at 6 groups of red-tails. And we tracked individual patterns of participation during these conflicts and collected urine in order to measure C-peptide of insulin, which is a byproduct of insulin metabolism and indicates how well off the animal is doing in terms of energy. High C-peptide is good, indicates a surplus of energy. Now, just looking at the relationship between C-peptide and participation doesn't really provide any clear pattern, but I did notice that there is a pattern where some females are conditional cooperators. Sometimes they participate. Other females basically never participate. These are what I call the non-cooperators. Now, even among the conditional cooperators, no female participated in every single conflict. So there's variable participation over time. And then what we see is that there's a fairly strong predictive power here where the non-cooperators have lower energy balance than the conditional cooperators. The conditional cooperators, for a particular fight where they happen to not be participating that day, they have very high energy balance, basically a surplus of energy. And I interpret this as meaning that they're doing well enough that increased access to food resources isn't really necessary. They already have that surplus. When conditional cooperators actually participate in a fight, they're in this Goldilocks zone where they could be doing better. So perhaps by fighting, they might be able to gain better access to food, but they're not doing so poorly that they can't afford to fight. Now, comparing post-conflict energy balance to pre-conflict energy balance, we see a slightly different pattern where in some ways, there's actually a little bit of evidence of a collective action problem. So a collective action problem is when there's a resource that benefits the whole group, but only some individuals are participating to defend access to that resource, and they're thus bearing the costs of participation. So here we see that non-cooperators are benefiting after an intergroup encounter and their energy balance has increased. So again, the defectors are sharing in the spoils of battle. This is fitting with the idea of a collective action problem. But the conditional cooperators, regardless of whether they're actually fighting or not fighting, are seeing a trend toward a decreased energy balance, although it's not substantially different from zero. And this is sort of suggestive of a collective action problem because it implies that there might be a cost, but it's not a great enough cost that it sort of differentiates between periods when they are and are not fighting. So I'm still sort of thinking about what this might mean in terms of the pattern of participation and motivations for participation. And the interesting thing here is that this is a pattern that holds up regardless of whether the group won or lost the conflict. So it doesn't matter how well the group did. And the issue here is that winning or losing in conflict is not what we think it means. The group that wins the conflict is the group that had been feeding there for some period of time and was willing to leave when the new group approached. So it's about satiation. It's not about dominance, at least in red tails. We also do see a pattern that indicates that there's some degree of energetic cost associated with these conflicts when we look at other biomarkers. So for cortisol, there's a slight increase in this stress hormone after a conflict, and then a slight decrease in testosterone. Again, this is congruent with a pattern where there's energetic outlay that occurs during these conflicts. However, this is a pattern that holds up regardless of whether the individual actually cooperated or participated aggressively in the conflict. So that part kind of bucks our expectations. Now, thinking about longer-term patterns and the costs and benefits of these conflicts, I wanted to look at how groups use that shared area of overlap between the home ranges and to determine whether there's a pattern of one group having priority of access to that area. And to start off with, we have to take into account the size of the overlap area. If the overlap is a very large part of the home range, it should be used more often than if it's a smaller part of the home range. And that's exactly what we see for both red tails and mangabeys. Red tails in the sort of orangey color and mangabeys in blue. So the R-squared values are quite high, and the larger the overlap area, the more often it's used by the group. Taking the residuals from these scatter plots and then looking for a particular dyad to see if there's a pattern, we see that for mangabeys, there's a strong negative relationship between how group 1 uses that overlap area and how group 2 uses it. So there is sort of a pattern, a dominance-like pattern for mangabeys. but not so much for red tails. It looks like there's a slight positive trend, but again, the R-squared value is a bit low. And again, red tail groups tend to bump into each other. They're not tracking how the neighbors are using these overlap areas. Now for mangabeys, what might explain why one group is overusing the overlap area and another group is underutilizing that area? There's no relationship to group size or to numeric odds here. Instead, what seems to potentially be happening, this is a little bit of hand waving here, I did see intergroup infanticide between some groups, 2 of my 6 study groups, and the consequence of that seemed to imply that the group that committed the infanticide had greater access to the overlap area after the infanticide. And the group that had experienced the infanticide seemed to underutilize that shared area of overlap. So it seems that the groups are negotiating their use of space through the threat of infanticide. Now, how broadly this occurs across primates, who knows? It's a very rare thing to observe. So hopefully moving forward, we'll be able to understand the patterning of this particular pattern of threat a bit more clearly. So I want to just point out that there's a large body of literature relating to evolutionary game theory and assessment strategies in animal contests. And a recent review sort of suggested that we move forward in slightly different directions. So Chapin et al. Indicated that assessment of resource holding potential does actually vary across individuals, contests, and phases. And that was kind of a new perspective. And they put forward this triangle where contestants monitor their own resource holding potential, certainly. They might monitor the opponent's resource holding potential. And then, of course, there's the resource value at the apex of the triangle, which should also affect their strategies. What I have found from the mangabeys, however, is that there's different resource characteristics that predict behavior at each contest phase. So that indicates that the value of the resource kind of is differentially assessed based on the intensity of the threat during the contest. And interestingly, there's assessment of the resource value not only to oneself, but to the opponent. So instead of being a triangle, I would propose that it's more like a square, where it's self-assessment of the resource value and resource holding potential, and other assessment of the others group's resource holding potential and the value of that resource to that group. Moreover, I mean, this is kind of exciting, right? The cooperation effect seemed to swamp the effect of resource holding potential for mangabeys. And clearly this is not necessarily a widespread pattern. We see the opposite in capuchins, but this is one way that groups might be able to sort of shift the balance of the asymmetries that we see in these contests. So there's a lot that sort of goes into these intergroup conflicts. It's not only the food spatiotemporal characteristics and group density and resource limitation and intersexual conflicts. And so food defense is not an inherent trait of the consumer. It's not a species-specific pattern, and it's not necessarily predicted by being a frugivore or folivore. It's subject to all these influences. And the energetic payoffs that we see do vary across individuals and affect their patterns of participation. So moving forward, I recommend that we would test diverse aspects of multiple contested resources, food and mates, for instance, and for food that we test both local and regional characteristics, and keep in mind that some motivations can cancel each other out, and to scale up from individuals to sexes to more fully understand group behavior. And with that, I would like to thank my staff, students, and collaborators, and thank you guys for your time.
Great. Thank you so much, Rochelle. That was really interesting. So I have a few questions. I'll start us off with one. So I was curious of the costs and benefits of the red-tailed monkeys for the red-tailed monkeys of traveling with the mangabeys. So like, why are they traveling with the mangabeys?
Yeah, that's a great question. So it's not one that I have studied directly, but Tammy Winfelder and Jerry Lawanga studied this around 1999, 2000. And they were finding that it seems to be about anti-predation benefits rather than, say, a foraging benefit. And the idea here is that mangabey males are very effective at countering some of these predators. So for instance, when they see a crowned hawk eagle, which is one of the main predators of primates in this area, the mangabey males will chase it away. And so that seems to be to the benefit not only of their own group mates, but to any other species that is associating with them. Now, of course, the other main predator in this area is chimpanzees. And I'm not so sure that manga bees are effective in that respect, but yeah.
Cool, thanks. And then the other thing I was interested in was the idea that the manga bee females aren't really participating in these intergroup conflicts. And so you showed in the red tails that it seems like maybe there is a cost to participation that's maybe not outweighed by the benefits. Do you see the same thing in the manga bees? Or so in other words, do all the group members in the manga bees get access or to food or resources, or do the participants get more?
So I haven't actually looked at that level of the question for manga bees yet, because that project was just on the red tails. But recently I've been acquiring some information. So maybe stay tuned, and in a few months I can answer that question. But yeah, at this point, I'm not sure. Great.
Well, I'm really interested to hear more about that. And I think we'll stop there since it's 1 o'clock. But I'd like to thank both of our speakers, Lauren and Michelle. And for everyone out there, we will see you next week for one last seminar of the semester. Have a great day, everybody.
Thanks.
Thanks. Bye-bye.